Right, that's why I had a pretty scary. Why do you look at, oh, I'm sorry. Yeah.
You tell me Montreal. That's a guy. I know that guy. Yeah. Dude, honestly, I like it. Yeah, I think otherwise. Yeah, I know, I do. I you're a friend from Montreal, and I don't like I know.
Yeah.
All right, everyone, we're a little past, but everyone is to remember to here, so we're officially, started. Thanks for coming to Kamron's defense. I'm not good at this, and I don't have photos like my most experienced and smart colleagues. I don't take pictures. But I have a few words. Kamron was a fantastic member of the lab who we set to see him eventually leave, hopefully not anytime soon, even though he's standing. But he arrived in the lab in 2018. He graduated from IB. He actually was an IB student in biology. He was very involved in undergrad research in different labs. And he joined the lab in the fall of 2019. He applied kind of late. And at that time, our lab had just received a new research grant. So I was very excited to recruit new students. I was a new professor here. And I was interviewing students, flying them in from all over the country to interview, and we were about to pull the trigger on one, and then we got an email from Kamron. But no, I actually work downstairs, I can just come up and tell you. Sure, why not? Then he comes, and then he walks in a couple days later with his long luscious hair and confidence of the Jedi, and I just stopped it. He talked to everyone in the lab independently, and I mean, by the end of the day, we decided on Kamron because he expressed it. One was the engagement, the level of questioning he had on maybe one project, even though they just that was like a true motivating work for us. So I think Kamron, and we're super happy that he did. So he started in 2018. He applied for the NSF fellowship. He got it first time. A lot of you are on this boat right now. He started fellowship, and then COVID hit. So right in the beginning of this project, we had to lockdown. But he got permission to brave the empty hallways of old world for several months. And he collected enough data during those quiet months to actually get his first chapter and publish that. Not too long after that, that was very good motivation. And then he started, he actually collected data for 2 big projects early in the program. But then the third one was very ambitious, took a long time. And I'm telling you this because he's going to undersell how difficult that third project is because they have time and that's his nature. But it was a very difficult, ambitious project that in our system, our code system, it's very difficult to pull out. It's only done a couple of times by other labs in the country because it requires 12 months of reading code in a very specific way. So he did it. He actually developed a really strong mentorship relationship with stellar undergrad and IB. And the 2 of them just hunkered down for 18 months and got pulled it off. So I'm really excited about the project, and I think it will be an impactful publication. And finally, I think we have a lot of people here because we know Kamron was a great member of the community. Very friendly, lots of cohorts, and he very friendly with everyone. He also was very involved in a lot of different levels of engagement in the university department level. He co-founded the IB committee within the department. He also co-founded the university EMUs, the ethnic minorities in STEM. That's a big group as well. So that's higher level. And then within the lab, we also do a lot of work with LSAM every year, and he's helped us run workshops for the undergrads at the LSAM group. So a lot of involvement. So I'll kind of leave it at that, and hopefully you enjoy hearing about some stuff. So thank you.
Thank you for the introduction. Is my mic okay? Can you guys hear me pretty well? Maybe I'll just speak up a little bit. Well, thanks for coming today, everybody, for my dissertation defense talk. Welcome. So today I'm going to be talking to you about the research I did for my dissertation, which is on looking at the role of mitochondria in local adaptation to thermal stress. And to get started talking about this topic, I'm going to explain what local adaptation to thermal stress is. And I'm going to do that by talking about, whoops, my clicker is not working. There we go. I'm going to start talking about allopatric populations to introduce this. So allopatric populations are populations of the same species that are geographically isolated from one another. So for example, if we have 2 populations of bunnies, population A and population B, they are geographically isolated, so they can't meet one another. Right, in this case, bunnies can't swim, so these individuals will never meet these individuals. And because they're geographically isolated, we can say that they have no gene flow. So they can't reproduce because they can't meet. So genetic information from population A will never enter into population B or vice versa, they'll never mix. If these two populations experience different temperature environments, for example, population A's environment is quite warm, and population B lives in a colder environment, they're going to be experiencing selection pressures from those two different environments that will alter the genetic makeup of their populations such that they should become adapted to different temperatures. So we would expect this population to have changes in the allele frequencies in its population to become more favorable to tolerating heat than the other population, right? And that can result in phenotypic changes between populations, right? So this process of local adaptation to their temperature environments increases genetic divergence between populations. It causes them to become more genetically distinct from one another. And over time, because these pressures are going in different directions, that could cause these populations to become so distinct that they might actually become different species at a certain point. So even if they met and reproduced, they wouldn't be able to produce viable offspring at that point. So this is local adaptation to thermal stress. Let's talk about where mitochondria come into that. So mitochondria are often considered to be the powerhouse of the cell. You've probably heard that before. The reason they're called that is because the mitochondrion is the site of a really important metabolic process for eukaryotic organisms, which is called oxidative phosphorylation, or OXPHOS. And so the mitochondrion is the site of OXPHOS, and what this is basically a process conducted by a series of protein complexes in the mitochondrion that we call 1, 2, 3, 4 ATP synthase. And to make a long story short, basically what they do is they allow us to use food and oxygen through a series of chemical reactions where we store energy in the form of a molecule called ATP. And so you need ATP to power all the chemical reactions in the body. And so if this OXPHOS process isn't working correctly, organisms will suffer losses in fitness. They will be less likely to survive and less likely to reproduce. The mitochondria is not just a simple structure though. So mitochondria are actually the result of a very ancient form of symbiosis. So long ago, the mitochondrion was an independent bacteria, an ancient bacteria, and it formed a relationship with an early cell. And over time, the two have co-evolved, and they descended into all the modern eukaryotic lineages. So basically all life that isn't sort of bacterial-like. And as a result of that, the mitochondrion actually has its own genome, which is distinct from the nuclear genome. So it's evolved to lose a lot of its functions over time, but it still encodes several proteins that participate as subunits of these larger complexes that we see in OXPHOS. So these are large complexes made of many proteins. Some of them are made by the mitochondrial genome, and the rest are made by the nuclear genome. And so the function of this process, OXPHOS, is actually the result of interactions between the two genomes at many of the complexes, and we would call these mitonuclear interactions. So for OXPHOS to be working normally and physiology to be working normally in an organism, both of these genomes need to work well together. It's no plant case. And besides OXPHOS, pretty much all of the mitochondrial functions are the result of interactions between the two genomes. Transcription and translation of the mitochondrial genome is done by components that are encoded by both. So if these two genomes don't work well together, basically all mitochondrial function will be impaired. The two genomes are quite a bit different, as you can imagine. So the mitochondrial genome, being the remnant of a bacterial genome, is actually haploid instead of diploid. It's usually, in most eukaryotes, it is replicated clonally and is inherited through the maternal lineage rather than through recombination of both parents. So you usually get your mitochondria from your mom. Thanks, Mom. And because it's also quite a bit smaller, all these factors contribute to it having a much faster rate of evolution typically. So small changes in this mitochondrial genome have larger evolutionary outcomes. Right? And when this mitochondrial genome changes, that can cause those OXPHOS subunits that it encodes to no longer match correctly to the ones encoded by the nuclear genome, which causes a reduction in OXPHOS function. And then that in turn causes organisms to survive less, and it selects on a population such that the nuclear genome shouldn't match to compensate. Right? We have to keep, every time this evolves, this must evolve to make sure OXPHOS keeps working. Right? So we call this process mitonuclear coevolution, and it's basically the selection pressure that acts on the nuclear genome as in response to changes in the mitochondrial genome at these OXPHOS locations. So if we go back to our locally adapted populations, we know that genetic divergence between them is a result of local adaptation for their different environments. But because they have no gene flow, it's also the result of each of them having independent bouts of mitonuclear coevolution. So this process also contributes to divergence between them. So I wanted to look at these two evolutionary processes and basically ask, how do they interact with one another? Is there ever evidence that they facilitate each other? Can local adaptation act on the mitochondrial genome, which then in turn would reinforce divergence? Or do they ever come into conflict? Is there ever evidence that mitonuclear coevolution could prevent local adaptation to thermal stress? And so these are kind of the broad questions that I wanted to explore. And to look at these, I use a study system called Tigriopus californicus, which is, I would say, about as cute as a bunny rabbit. This is a harpacticoid copepod, which is a crustacean. It's native to North America on the west coast, and it lives in these tide pools. So here's a picture of a female and a male as a pair, and then this is an adult female with a clutch of eggs. And this is a picture of a tide pool that they live in. And if you look closely, each of these little specks in the water that looks like dust, this is actually an individual organism. So each speck is a red bud. So they live in large populations in these high tide pools. And these tide pools are called supralittoral, which means they're above the high tide line. So when the high tide comes in, it doesn't actually reach them. So they never get flushed out and mixed. And as a result, the populations that are at distinct places on the coast don't ever come together. So they pretty much have zero gene flow. Each of these populations is very isolated from one another because they're trapped in these tide pools, basically. These populations are clearly allopatric because of this, right? And they also show evidence of both local adaptation to thermal stress. So this plot up here, the red graph, is looking at tolerance to temperature at different latitudes. And we can see that as we go north, populations are less tolerant to high temperatures. So southern populations, like you might expect, they live in warm environments. They're locally adapted to tolerate higher temperatures. We also find really high levels of mitochondrial sequence divergence between these populations. This plot's showing divergence in the mitochondrial genome at several different genes, and the little bars are comparing multiple populations. You don't have to look at it super closely, but basically what this is saying is populations that, because they don't mix, they have divergences in the mitochondrial genome of 20 to 30%, which is huge. That's about a quarter of their mitochondrial genomes are different. And this difference in mitochondrial genomes isn't just neutral, it does have selection effects. So if we created hybrid populations of these allopatric populations, over time, hybrids can experience what are called mitonuclear incompatibilities. And this is basically the result of OXPHOS subunits encoded by the mitochondrial genome, which we're showing as a little ring here. If they're mostly from one population and they don't match most of the ones encoded, say, from the other population, that causes loss in OXPHOS function, which causes individuals to be less likely to survive or reproduce. So this is a very serious selection pressure that reduces the ability of these populations to hybridize. So I chose two populations, the Strawberry Hill population from Oregon, which I'm going to call SH. I'm usually going to show this little blue copepod, and the San Diego population, which I'm going to call SD. And as you might expect, the Oregon coast population, not particularly used to high temperatures. It's usually pretty cold. So this population has a much lower thermal tolerance than this one. The California population is more tolerant. So SD is more tolerant. And they also have really large amounts of documented genetic divergence. The nuclear genomes between these two populations are about 2.8% divergent. That doesn't sound like a whole lot, but that's actually more than humans are from gorillas. So these two populations are quite distinct from each other. And then their mitochondrial genomes have an average divergence of 23%, so huge, about a quarter of their genomes are different. This suggests that both of these selection pressures that I'm interested in apply to these two populations. And so I wanted to use them to examine kind of three research questions. One, does mitochondrial genetic variation actually contribute to differences in the ability of individuals to tolerate temperature, the phenotype? Two, does mitonuclear coevolution actually act on these populations and change how much they use their genes? So that's gene expression. And then the last one is, does my nuclear coevolution ever actually come into conflict with selection? Does it ever prevent selection for thermal tolerance? And I addressed these questions in my 3 dissertation chapters. I'm going to go through them one at a time. So I'll start with the first one. So in this chapter, I was looking at physiology of the organisms. So I was looking at whether differences in mitochondrial genomes actually contribute to differences in physiology of tolerance. There is some evidence from previous studies to suggest this could be the case. So multiple studies have found evidence that mitochondrial genomes between populations show differences in sequence based on the temperature that those populations experience. And a few studies like this one by Camus actually showed that in Drosophila flies, there was a difference in organism tolerance depending on mitochondrial background. So this suggests that their differences in mitochondrial background could contribute to local adaptation. In Tigriopas, a couple of studies have looked at mitochondrial function, that potential for local adaptation and function. So this is showing ATP synthesis rate. So this is mitochondrial function, the function of OXPHOS at different temperatures. And what they found is different populations show different amounts, so different rates of ATP production, and also different stabilities of rate. The temperature affects that rate more or less, depending on the population. So this could be evidence that local adaptation is happening in mitochondrial function. However, we know that OXPHOS is actually the result of interactions between the 2 genomes. So maybe it's local adaptation here, or maybe it's local adaptation here. We need to parse them out. And further, tolerance to heat is actually usually the result of these proteins called heat shock proteins that often are involved in like stabilizing molecular reactions that are under stress. And most of those are encoded by the nuclear genome, along with about 1,000 other proteins that are actually inside of the mitochondria while it's working. So we need to parse out, is it the mitochondrial genome or is it the nuclear genome that's causing this local adaptation and function? To do this, I took our 2 populations, SH and SC, and they have different nuclear genomes, and they also have different mitochondrial genomes. And I created 1st generation reciprocal hybrid crosses between them. So the naming scheme here, this is SH mom and SC dad and reciprocal. And in T. californicus, 1st generation hybrids are heterozygous, which means they have about equal contribution to their nuclear genome from both parents. So they have very similar nuclear genomes. However, mitochondria are inherited matrilineally, so they get their mitochondria from their mom. So the difference between the crosses genetically is mostly the difference in their mitochondrial background. So they have whatever mitochondria their mom had. So I use these 4 populations to test 2 measures of thermal tolerance. 1, survivorship following stress, so can you actually survive stress? And 2, stability of ATP production. So does ATP production break down at different temperatures? I'm going to go through them one at a time. So I'm going to do the methods and the results for both of them separately. So to look at survivorship, I took groups of 10 individuals at a time per genetic background, and I put them in a hot water bath and stressed them out for an hour. And then 3 days later, I checked the survival proportion of those 10 individuals. And I repeated this 128 times per genetic background. And when we think about expected results, so if we thought about, okay, the percent survival of that group of 10 at different temperatures, we know that SD is more tolerant than SH. So we would expect that the SD population is going to survive better than the other one. SD should survive better than SH. And if mitochondrial genetic background is contributing to this, we would expect that the hybrid cross that has the SD mitochondria is going to survive better than the other cross, right? Because they're not particularly different in their nuclear genomes. They have different mitochondria. So this is what we would see if mitochondria is contributing to that difference. When we look at our actual results, we don't quite see that. We do see clear evidence that SD is more tolerant than SH. So yes, they are locally adapted, confirmed. But when we look at our hybrid crosses, we actually find that they are both intermediate to the parents, and they weren't statistically different from one another. So they have the same survival, regardless of mitochondrial background. And this suggests that mitochondrial background doesn't really matter. It's not really contributing heavily to whether or not they can survive. And further, that the differences that we see between SH and SD are probably the result of differences in their nuclear genomes, because our hybrids, which are heterozygous, have about intermediate. So they're 50-50 in the nuclear genome, and they're not right down the middle in terms of tolerance. So no evidence of differences based on mitochondrial background for whether or not they can survive. When we look at stability of ATP synthesis, so mitochondrial function, What I did is I took groups of 24 individuals of each genetic background at a time, and I isolated their mitochondria. And so this is a picture of what isolated mitochondria in a group looks like. They look like a brown speck. And so I took that pool of mitochondria and I split it evenly between 5 temperature treatments. And then I ran them in the thermocycler. So what this lets me do is basically look at each of these temperatures, what was the rate of ATP production? And then I can compare how much that rate changes between temperatures, and that's stability. So if we think about our stability over temperature, we should look at our ATP synthesis rate at different temperatures. And we know SD is more tolerant than SH, so we would expect that SD's rate is more stable. It should change less as temperature changes, so it should have a flatter reading. The SH population is less stable or less tolerant, so we would expect it to be less stable. It should be a little bit more erratic. As temperature changes, there should be big changes in ATP rate. And then if that's due to mitochondrial background, we should be kind of similar to tolerance. We would expect that the cross that has the SD mitochondria should be more stable than the other cross. This is what we'd expect to see if mitochondrial background is contributing. When we look at this, again, it's not quite what we expected. Yeah, we do see clear evidence that the SD population has the most stable rate. It was statistically the most stable. It changed the least as temperature increased. And the SH population in dark blue is quite a bit less stable. But when we look at our 2 hybrids, we again find they have actually very similar changes in rate as temperature increases, and both of them are very unstable. So this is suggesting that mitochondrial background doesn't really play a big role in stability. And so to summarize this first chapter, when I was looking at 2 measures of thermal tolerance, survivorship, the hybrids were intermediate to the parents and not different, suggesting mitochondrial background didn't contribute to actual survivorship. And similarly, while SD was the most stable population in terms of ATP rate at temperatures, the hybrids were about the same as well. They didn't show a difference based on mitochondrial background. So no difference in physiology. So I'm going to switch over to my second chapter now. And so this is focusing on whether mitonuclear coevolution actually contributes to differences in how genes are being used. So the amount that genes are being expressed in different populations. And when we think about phenotypic divergence, so like differences in whether or not they can tolerate heat, right? That can be the result of differences in the structure of proteins. So maybe one population has a heat tolerant protein and the other doesn't. Or it can be in differences in how much they make those proteins, the differences in how much they express them. And this is what I'm going to focus on. So this study is looking at differences in expression between these populations. Specifically, how does mitonuclear coevolution, or how may have it affected the evolution of differences between SD and SH? So when we look at some previous studies, we have, if we're looking at hybrids of SD in the population called Santa Cruz, there's some evidence to suggest that both evolutionary processes could be at play. So this figure is, don't worry about the graph too much, but the gray dots are representing genes that are upregulated in hybrids that are healthy and fit. And those genes included ones that are involved in oxphos. So that suggests mitonuclear coevolution could be driving their increased expression. However, when we look at those same hybrids under stress, there is evidence to suggest that they tend to express their genes the same way that the more tolerant SD parent does. So these red dots are representing genes that are being expressed in the hybrid the same way as SD expresses them. So this is suggesting that local adaptation is driving how those genes are being used. So there is some evidence to suggest in C. californicus that both mitonuclear coevolution and local adaptation to stress contribute to differences in how they use their genes. I want to compare this a little bit more directly, so I set up a test to kind of look at them side by side. And what I did is I took our same 4 populations, SD, SH, and the 2 F1 hybrids, and I took groups of 20 individuals at a time. So this is like a tiny little pod here, and there's 20 here. And I gave them either a stressful treatment in a hot water bath or a control treatment in a cool water bath. And then I extracted RNA from them right after that treatment. And so when you extract RNA, you get basically a snapshot of what gene was being expressed, what expression looked like at the time. So we can look at these 4 different crosses, what was expression like when they were under stress or under control. Then we compared and sequenced them, which let us basically compare them to a reference called mapping. And this is a little bit of an abstract concept, so I'm going to explain it using an analogy. So the idea behind mapping is each sequenced piece of RNA we got, you can consider it to be kind of like a puzzle piece. And you might hold up a puzzle piece to like the box that the puzzle came in to figure out does this piece go in this box or did it come from this box, right? You can also figure out what part of the puzzle the piece is supposed to be, right? And so what we did is we took our sequence pieces of RNA and we held them up to both reference genomes. So this is the sequence of the entire population. And we can figure out, okay, does it come from this one or does it come from this one? And what gene is it? And so if we get a bunch of these, we can say, okay, this gene was upregulated a bunch of times in this population or the other. So we compared the sequences we got from our two parent populations. They typically map to the correct population when they're given the option. So we gave them both options. They map usually where we would expect. And then we look at our hybrid populations. Because they're heterozygous, they produce both kinds of sequences. So we gave them a hybrid reference and we let them map where they were most appropriately mapping. And so what we can do with this is we can basically compare which population was being mapped to, how many times the sequence was being used, so that's the amount of expression between these two crosses to see if there's a difference between them. We use this kind of approach for we use this mapping in two approaches. One, we looked at parental dominance in expression, which is basically asking, does each hybrid express their genes the same amount as the SD population or the same amount as the SH population? And then we also looked at allele-specific expression, which is basically asking, does a hybrid preferentially use the SD version or the SH version? So this is amount, do you behave like one of your parents more, or do you preferentially use one of your parents' versions of the allele? So when we think about the first method, I'm going to go through them one at a time again. This is looking at parental dominance. And so what we can do is we can infer inheritance of the expression. And there's a bunch of different patterns that we can look at when we compare the amount the hybrid expressed the gene versus each parent. I'm just going to focus on these two today. And so we've got two cases, SD dominant or SH dominant. In the first one, if the hybrid's expression in pink is very similar to that of the SD parent, but statistically different from that of the SH parent, we can say that the hybrid behaves like SD for this gene. So the gene is expressed the same amount or the same way as SD would express it. So we would call it SD dominant. And then the reciprocal is the case when it's SH dominant. So we're looking at comparisons between the amount of times it was SD dominant or SH dominant, how many genes fell into each of those categories. So if we look at expected results, we're looking at the number of genes that fall into each of these categories in each of these crosses. And what we should expect to see is, or I guess I'm going to break down what we'd expect to see if either mitonuclear coevolution or local adaptation to stress is sort of driving the ship about how expression is being used. So if mitonuclear coevolution is prominently driving regulation, we would expect more maternally dominant expression. So more genes should be expressed similar to the maternal population because mitochondria are inherited from the maternal population. So we want to match, they're sort of adapted to match the same way that the maternal population does it. So it would look something like this if mitonuclear coevolution is responsible for the differences in expression. If local adaptation to stress is driving it, both populations should favor expression similar to SD because we know SD is the more tolerant population. When we look at our actual results here, I kind of blew them up here to make it easier to see. If we look at the same crosses under control conditions only, we do see evidence that maternally dominant expression is more common. So this cross that has the SD mom has more SD dominant expression. This cross with the SH mom has more SH dominant expression, which is cool. This is actually evidence that mitonuclear coevolution could be driving baseline expressions between these two populations under control conditions. When we compare that to under stress, however, we see something a little bit different. In this cross, SD dominant stays the most prominent case. And in this cross, however, they become almost equal. So SD dominant expression increased in this cross under stress. Right? And this is suggesting that under stress, expressing something more like the way that SD expresses it is actually favorable. It's a response to that stress. So this is suggesting that SD dominance is prominent, that local adaptation is driving expression differences under stress. So we found evidence of both. I took it a little bit further. I wanted to kind of parse them out a little bit more. And so if we look at our cross here, it has an SD mom, it's under stress. So it has a bunch of these 2000 genes that are SD dominant. This could be the result of either process though. This could be the result of local adaptation to stress, or because it has a SD mitochondrial background. And so what I did is I took the 2000 genes that are in this group and I identified their functions or identified their names basically, and I matched them to where they show up in this cross to see whether the pattern stayed. And what we see is about half of those 2,000 genes are also SD dominant in this cross, suggesting that local adaptation is a driving force in about half of those genes. And then about 14% of them are SH dominant in this cross, so maternally dominant, suggesting that more of them show a response to local adaptation distress than to nuclear coevolution. Okay, that was the first part. Second part, we're talking about allele-specific expression. So we're talking about whether the hybrid is preferentially using at each gene the SD version or the SH version, which version is favored to be used. And thanks to Yang D for his help on this part statistically. So to think about this, we're thinking about for a given gene, so just pick one gene, we look at the control conditions. If we have more times that a sequence piece of RNA matched to the SH reference, we can say that we have SH bias. So the allele for SH is being biased towards under control conditions. And then if we look under stress and we see that there's a big change, for example, a lot more SD is being used under stress, we can say that bias changed towards SD under stress. And so we did this for all the genes that we found evidence of some kind of bias. Expected results for this, we're looking at the number of genes that show bias to each parent under control conditions by cross. And so we can think of the height of the bar here as how many genes were biased toward that parent's allele under control conditions. Like you might expect, if mitonuclear coevolution is causing preferential use, we would expect more bias towards the maternal population under control conditions. And then if we think about how these genes change under stress, if local adaptation is driving it, more of them should change towards SD than towards SH. So more orange than blue, basically. When we look at our actual results, we see something kind of weird, which is cool. We found about 900 genes that show evidence of some kind of bias. But when we look at our two crosses, we see a lot of evidence of SD bias. So using the SD allele more often in both crosses under control conditions. So the SD allele is biased towards regardless of mitochondrial background, which could be evidence of local adaptation distress. The SD allele maybe is just preferentially used. These are under control conditions. And then when we look at under stress, both crosses actually change more towards their paternal parent. Right? This one's changing more towards blue. This one changes more towards orange, suggesting that they use their dad's allele more often under stress than either the maternal allele or SD. So this doesn't really provide a ton of support for either of them. Right? It suggests that SD bias is common under control conditions, but neither process seems to be driving bias under stress, which is kind of cool. So to summarize this chapter, we did find evidence that both evolutionary processes are contributing to differences in expression between these populations. And when we look at our two hybrids, maternal dominance is prominent under control conditions, which suggests mitonuclear coevolution is playing a role in how genes are being used. And under stress, however, we do find evidence that SD dominance is more prevalent, suggesting that under stressful conditions, it's beneficial or local adaptation has driven use of SD. But when we look at allele-specific expression, so which version do you use, we find that the SD allele is more preferentially used under control conditions. It's maybe an evidence of local adaptation, but under stress, it kind of follows in either pattern. So something to explore further. Okay, gotta get a sip of water. Moving on to the last chapter. Okay, so in the last chapter, what we wanted to do is compare directly these two selective pressures. I wanted to see whether mitonuclear coevolution could ever prevent adaptation to thermal stress. And if we look at our two locally adapted populations, right, we know one of them has much higher thermal tolerance than the other, and that is a result of differences in their genomes, right? The response to being selected for being tolerant. So they have different genomes or genetic differences that contribute to this difference. If they ever find each other, if individuals from this population meet, they can reproduce and produce hybrid offspring, which can introduce information from this population into this one, right? And if those alleles for tolerance to temperature enter into this population, it can improve this population's thermal tolerance, right? This is called adaptive introgression. The alleles are adaptive, they introgress into this population, and if they're under stress, they become more tolerant. So this can be a really useful sort of evolutionary process for rescuing populations that are not particularly sensitive, so like in the face of climate change. Hybridizing populations is one way to improve their tolerance and kind of rescue them from the rising temperatures. This evolutionary process can be impeded by a couple of mechanisms though. For example, if the alleles that are associated with thermal tolerance are also maladaptive in the new population in some other way. So like, for example, maybe they give you thermal tolerance, which is great, but they make you less likely to reproduce, like they make you smelly and ugly or something like that. Because they kind of come together, right? This allele gives you thermal tolerance that makes you less likely to reproduce. There's a conflict between selection, and this allele won't become more prevalent in the population. It has to deal with that conflict. Similarly, if those alleles are linked physically to genes that are maladaptive, so the thermal tolerance allele is physically attached to a gene that makes you smelly and ugly, right? They always come together as a package deal, and so there's still a selection conflict between them if they're linked. And so I know what you're going to ask at this point. You're going to ask Kamron, what does metanuclear coevolution do in this process? Can this be a part of the maladaptive or linkage to maladaptive? Can it prevent adaptive integration? And I'm going to try to answer that for you today. So we know that oxphos function is the result of interactions between our two genomes, mitonuclear interactions. And we know that if those mitonuclear interactions go badly, we get incompatibility, which causes losses in fitness, like death or inability to reproduce. And that this is maladaptive, so dying is maladaptive. What you get here basically is if alleles in a hybridizing population are selected for, say it's hot out or the temperature is hot and for the hybrids, if alleles give you thermal tolerance and it's hot, they're going to be selected for. They should increase in the population, but if they are linked to genes that are involved in mitonuclear incompatibility, they'll be selected against because they cause you to reduce your fitness through those incompatibilities. And so basically, we get a conflict here, and I wanted to examine who would win. So hybrids in previous studies between SD and a population called Bodega Bay did show that adaptive integration was possible after a selection experiment. So hybrid populations of those two that were experimentally evolved to be tolerant to heat were able to, and that was related to an increase in the SD allele in their populations. So becoming more SD made them more tolerant, suggesting that adaptive integration can happen in this system. And I'm going to take this a little bit further. I basically wanted to ask, is there a difference in how much that adaptive integration process works based on mitochondrial background? Does metanuclear coevolution compete with that selection process? So I took my SAM crosses that we know and love at this point, and I split them into two batches. One that's selected for heat tolerance, so they were stressed out, and one that was under control condition for each cross. So we made 4 genetic groups. And when we think about these 4 groups, they're experiencing different kinds of selection pressures from the temperature stress and from mitonuclear incompatibility. If they're under selection for thermal tolerance, we would expect them to increase in their proportion of the SD allele because SD is more tolerant. However, we know that all 4 genetic backgrounds are experiencing selection by mitonuclear incompatibility, which should favor their maternal background. Right? So the heat selecting lines should favor SD. All crosses should favor whatever their maternal background is. And this group allows us to examine a conflict between them compared to all the other populations. So we can use this group to see basically who would win of these 2 processes. So we set up an experimental evolution experiment, and I had a ton of help from Jasmine Weaver, who is an undergraduate working in our lab, and now she's going to be a grad student at University of New Brunswick. So woohoo, she's awesome. And she helped me a ton. So we set up 6 experimental populations from each treatment, and we selected them over generations by exposing them to a stressful heat and then keeping all the survivors. So over time, we selected them for thermal tolerance for 10 generations. We gave them 1 generation to recover their population sizes. And then we used the last generation F12 for all of our tests to see what happened at the end, basically. In our controls, we did the same thing, except for rather than selecting them for thermal tolerance, we just randomly took out a bunch of individuals in the same proportion as those that were dying from heat. So this way, we're still removing the same amount of genetic information, but it's not due to response to heat. It's just due to like random drift, basically. And this was a huge project. This took, like Felipe said, this took 1.5 years of work every single day. And I wouldn't have been able to do it if Justin wasn't helping me in the lab every single day. A lot of beakers, a lot of cookie pods, a lot of counting. It was a ton of fun, but yeah, if you have a year and a half, feel free to do it. So at the end of that fall generation, we tested 4 responses of the selection. One, survivorship following stress. So do they survive better after selection or not? Stability of ATP synthesis, so differences in mitochondrial function. Life history traits, so differences in whether they reproduce, can survive to develop into adults. And then also changes in their genetic makeup, so changes in the allele frequencies. For the sake of time, I'm just going to focus on these 2 today. So I'm going to start by talking about expected results for differences in thermal tolerance, so the phenotype, the physiology. So when we think about thermal tolerance in each of our 4 groups, we have to consider the selection pressures they experienced over the course of the evolution. Our 2 controls, I'll start with them. They're only experiencing selection favoring their maternal background. So this cross with the SD mom should favor selection for SD because of their coevolution, and it should become a little bit more tolerant than reciprocal cross. And then when we think about our selected lines, both of them are experiencing selection for thermal tolerance, so they should both have higher tolerance. However, the group that has the conflict, we might expect them to have a slightly more mild increase. There's a conflict there that might be slowing down their ability to become tolerant. When we look at our actual results, up top here, we have average thermal tolerance. And then down here, we have each population individually that makes up that bar. And what we see is our 2 selected groups in yellow and blue, they did evolve. They do have higher tolerances than the controls. It worked. They're not different. They don't have any difference based on genetic background or based on mitochondrial background. So their tolerances were statistically the same. And then when we look at our controls, they're both lower and they're also not statistically different from one another, which suggests that mitochondrial background actually didn't impede the evolution of thermal tolerance. If you were selected, you became more tolerant. If you weren't, you were less tolerant. It didn't matter what your mitochondrial background was. So that's the actual phenotype. To look at changes in the genome, so changes in the allele frequencies in these populations, we sampled individuals from each of our replicates at the end of the experiment in that last generation. We isolated DNA from them, and then with the help of Dr. Molly Burke, we did library preparation and we sequenced them. And we kind of mapped them similar to before, so we mapped them against our hybrid. We saw our hybrid reference, we saw which of the sequences landed. And then we kept only single positions along the entire genome that differ, that are one base pair that are always different between the 2 populations. So in one population, it's always one way, and in one population, it's always the other. That single position is always different. These are called single nucleotide polymorphisms, or SNPs. And basically, we can use SNPs because they're always diagnostic. We can always tell which population they came from. We retained 2.4 million SNP positions to examine, which was a lot. And then we took those 2.4 million SNPs, we modeled differences in allele frequencies based on whether or not the group had been evolved by the selection treatment. And then we looked at each of those positions, are they more SD or are they more SH, depending on what group they're from. We got a lot of information, which is really cool. And basically what this plot is showing is each point is a SNP. So each point is a position in the genome. They're color coded by the chromosome that they come from. And the height of the point is the significance of the effect of the selection treatment. So if the point is really high, it means that the groups that were under selection have a very different allele frequency than the groups that were under control. We just kept the top 1% of them as a threshold because these are the regions that we determined are like heavily under selection. They responded a lot. They were the most significant. So we kept 24,000 SNPs to look at for our further analyses. When we think about changes in allele frequencies, it's going to be kind of similar to looking at tolerance, right? When we think about the selection pressures that are acting on them, and we can think about it in terms of how it changes the SD allele frequency. So how much SD was there? If you're over 50%, you're more SD. If you're under 50%, you're more SH. And so our controls, they experience selection by mitonuclear coevolution, which should become more like whatever their maternal background is. So this group should become more like SD, this one more like SH. And then our selected lines, we know SD is related to being more SD is related to being more tolerant. So we would expect them to both increase in their frequency of the SD allele. However, they go to the conflict again, should maybe have a more moderate increase. Right, so this is what we might expect to see if the 2 processes are in conflict. What we actually see is a little bit different, which is cool. So when you think about our two controls first, we do see that they became more like their maternal population. Their allele frequencies shifted towards their maternal parent, which suggests that mitonuclear coevolution actually does act on these allele frequencies that are involved in response to stress. So these are all the positions that were evolving in response to being stressed. And it looks like mitonuclear coevolution actually does act on them in these controls. When we look at our selected lines, things are a little bit different. They both increase in their SD allele frequency quite a bit. Okay? And actually the group that has the conflict increases the most, right? And so this is actually suggesting that the conflict is not strong enough to prevent increases in SD allele frequency. Actually, the stress treatment to high temperature is driving the evolution much more, right? So we did find evidence that mitonuclear coevolution acts on these positions that are under selection, but it's not sufficient to prevent adaptive integration. So to summarize, we're looking at tolerance in the organisms. The tolerance phenotype evolved regardless of mitochondrial background, so mitonuclear coevolution probably didn't impact it too much. And then we look at the actual genomes of these hybrids, we see that mitonuclear coevolution does act on these positions, right? It could be considered. However, under stress, the response to stress is a stronger evolutionary process than mitonuclear coevolution. And so just to briefly conclude about the whole thing, right? We didn't find any evidence of genetic background differences in mitochondrial background contribute to differences in physiology. However, we did find strong evidence to suggest that mitonuclear coevolution actually does impact differences in how the genes are being used between populations, and that it does act on positions that are under selection for stress. And this kind of suggests that, I think you should consider mitochondrial divergence between populations when you do things like planar reintroduction, especially for conservation. If you're going to hybridize populations, divergence between mitochondrial genomes can contribute to a selection process that could impede that. However, our third chapter really does suggest that adaptive integration, so increasing tolerance by hybridizing populations, could be a really strong way to rescue sensitive populations as temperatures increase with climate change. And with that, I'd like to acknowledge some people. So I'm just going to take a few minutes to have time to do acknowledgements.
We have a class apparently coming in, so yeah.
Well, they're gonna wait. They're gonna wait. So first and foremost, I'd like to acknowledge my advisor, Felipe Barreto. Thank you for all the mentorship and guidance that you've given me over the years. You really taught me how to be a scientist, which I really appreciate. This picture of us is actually staged. This is a stage photo. We're not doing anything here, but the photo is staged, but the mentorship was really real. So. I'd also like to thank all the members of my academic committee, Molly Burke, Yen Mindy, Jennifer, and Eduardo Cutia Sanchez. Thank you for all of your advice, working as my collaborators, and helping me refine and improve this work. I'd also like to give a big shout out to the Barreto Lab members, past and present. I just feel so fortunate to work with such an amazing group of people. Yeah, I really value you as friends, as colleagues. You helped me make my work better. I had a great time spending time with you, obviously, and I'm really excited to see all of your successes going forward. I'd also like to give a big shout out to some people in the department who made my time here really positive. The teaching teams that I was really fortunate to be a part of while I was here. The anatomy team, Devin, Lindsay, and Stacy, and also the introbio team, Alan and Lori. Thank you all for just helping me improve my ability to teach, being a mentor to me, being such great friends to me. And I'd also like to thank the office staff, Tara, Teresa, Tracy, and Melissa. Thank you for making sure that this day happened today, making sure that I could graduate, making sure I was registered for classes. I think I was actually late to register every single term. So thanks for making sure I was actually enrolled and that I had a job. I'd also really like to thank my amazing family who showed up today, and they've always showed up for me. You have always been my biggest cheerleaders. I really appreciate you all. I feel very fortunate to have you in my lives, you in my life. To all of my amazing friends that I've made over the years, thank you for your unending support, for making me laugh when I needed it, and for being there to tell me things about me when I wasn't really able to do that about myself. I really appreciate all of you, and you all had a really positive impact on me over these years. And last but not least, I'd like to thank my amazing boyfriend, Willie Curtis. I love you so much. I really appreciate you. You're a really bright spark in life, and I really appreciate all the support you've given me. Lastly, I'd like to thank National Science Foundation and Oregon State University and the Department of IB for funding. Thanks for the money. And with that, I'll take it.
Exactly, the project, one or two questions.
Making good, making easy. Yeah,
So you cross these warm and cold adapted populations. Then what temperature did you hold them at in the lab? And do you think that developmental plasticity could have had any impact on like their tolerances?
yeah, that's a good question. So we kept them under common garden conditions at about 20 degrees, which is sort of a not stressful temperature for either population. Plasticity between them, there have been a couple of previous studies that examined plasticity between T. californicus populations. I don't believe there's particularly strong evidence that plasticity is prominent in these populations. We also don't find a ton of evidence that individual populations had a lot of standing genetic variation that could contribute to adaptation. Yeah, that's a good question. I haven't really factored into phenotypic plasticity, but I don't believe it's a particularly strong force in the system, based on what I've read. Yeah.
What's next?
What's next? I'm gonna sleep for. I'm gonna take a break and figure out what comes next, I think.
All right, thanks, Karen.
And we're.